Hi, this is Mary from Open Helix with this week's Tip of the Week. If you're seeing this tip someplace else, be sure to swing back by our blog at blog.openhelix.com for additional details. I'll provide links to the resource that I'm describing today, links to their publication, and additional tools as well. So check us out at blog.openhelix.com for more. Today I'm going to be talking about FancyGene. And I was inspired to do this tip by a question that someone raised at BioStar. So the question was, how can I diagram my gene of interest? It has 60 exons and a whole lot of variations, and I want to do that accurately. One of the recommended tools to do that was FancyGene, so I checked it out, and here we are. Now, FancyGene will let you do these kinds of things, and it's a very simple interface. All you have to do is structure your data a little bit, click Draw, and you'll get output in various ways that you can use in seminars. You could use it in grant submissions. You can use it in publications. There's all kinds of ways you could use this. So FancyGene will let you do really complex diagrams with specific and accurate representations. So I'm going to tell you quickly too about a couple things you should check out if you're going to use FancyGene. Click their Help tab here to learn more about how FancyGene works. And they will give you some guidance on the input formats and how that data will be represented. But also be sure to check out their tutorial that has a lot more specifics on things that you might want to represent and how the shapes and the color codes and all that other stuff will work. So be sure to check out both the help and the tutorial to learn more about how to accomplish what you need to. I'll go back to the top page though, FancyGene, and I'm going to show you how I would illustrate this with a gene that exists in the UCSC genome browser. But you could do this later with your own genes of interest. They don't have to already be public information for you to use FancyGene. I'm just going to show you quickly how I might do this for a gene of interest. So let's say I was interested in the clock gene. This is the gene that Trey uses as his example in the table browser tutorial that we have, so I thought this was a nice one. If you don't know how to use the table browser to do this, be sure to check out our tutorial on that. But let's say I wanted to diagram out the features of the clock gene. I can do this by selecting the clock gene here. It provides me with a position. And what I can do is download the features in that position in gene transfer format by selecting output format here, click get output, and here's all the detail that I need. So what I can do here is select all, copy, go back to FancyGene, and paste this in. So all that information that I just quickly obtained from the UCSC genome browser, it will detect the right format here, or I could choose GTF in this case, and for now I'll just leave this as auto. It will detect that. Now you could upload this data if you had this in a file, or you could, if you had an ID, you could specifically retrieve that from some of these assemblies that are available. But for my purposes, I just wanted to illustrate how you could do it with a query. Now you could output this in different ways, as you can see here. You can do it in different orientations, and you can set border features if you want. You can do this later as well, but for now this is just a simple way to do that. I'm going to leave everything else as default. I will click the Draw button, and very quickly FancyGene will take that information and diagram out this accurate representation for me. So that's really handy, but now from here you can actually adjust this in further ways. So there's other things that you can do here. One of the things, for example, is you could change the shape. So I'm going to change the UTRs, for example, to oval, do a little bigger, and you could adjust the color code if you wanted to. I won't right now, but what I'm going to do is then go back here and redraw this with the new variation that I indicated, and look, now my UTRs are a different representation. So that's the kind of thing that you can do very quickly. In addition, you could add further features. So now, for example, if you wanted to add your variations, or you wanted to add domains or some other things, you could add features with the different options that you have from FancyGene that I told you to check out in the tutorial. It can help you understand that. So there's a lot of things that you can do at FancyGene to quickly and accurately draw gene structures and other features that you might be interested in representing using this terrific tool. And you can check out the citation for the paper that's associated with this here, and also you can learn more about the lab that's associated with this tool. Here they are, and check them out. This tool, FancyGene, is available for you to draw all kinds of features of interest on genes that you might be interested in representing. Thanks very much for your time.